To begin, let us acknowledge that Queen's University is situated on traditional Anishinaabe and Haudenosaunee territory. We are grateful to be able to live, learn, and play on these lands. We thank the Indigenous peoples of this area for their care of this land for thousands of years, and we hope to honor and respect them as we hold our virtual event today. I'd also like to take a moment to personally thank everyone for attending our presentation today. Our speaker this afternoon is Cyril Akwo, who will be presenting on spatial and temporal patterns of the black-legged tick, Ixodes scapularis, and Lyme disease in Ontario, Canada, making a cause for One Health surveillance approach. Cyril is a PhD candidate in epidemiology and One Health in Dr. Katie Clow's lab in the Department of Population Medicine at University of Guelph. He holds a Bachelor of Science in Microbiology and Medical Laboratory Technology and a Master's of Science degree in Medical Microbiology and Parasitology from the University of Biha, Cameroon. He also holds a Bachelor of Applied Science and Public Health from Ryerson University, Toronto. Cyril has research teaching experience and an interest in vector-borne diseases, clinical microbiology, infectious disease epidemiology, and foodborne zoonosis. Before joining Dr. Clow's lab, he was a food pathogen research associate at the Animal Disease Research Lab at the science branch of CFIA in Ottawa. He later worked as an assistant public health inspector at the Region of Peel Public Health Service. His current research project aims to develop and evaluate a One Health approach to surveillance of ticks and tick-borne diseases in Canada. This project recognizes the intrinsic and complex relationship between human, animal, and environmental health. It seeks to explore the patterns and distribution of ticks and pathogens, focusing on the black-legged tick and Lyme disease. We will have Cyril present, and then we will open for questions from the audience. You can ask questions by entering your questions in the chat box, raising your hand using the icon, or if time allows, unmuting and asking your question directly. Please help us welcome Cyril to the podium.
Thank you very much, Veronica, for that wonderful introduction. So I'm going to go ahead right now and share my screen for the presentation. And can everyone see my screen clearly?
Yes, we can.
Okay, so thank everyone for being here today, and I'll be sharing some findings of the work I've been doing for the past couple of years. And this work will be looking at the spatial and temporal patterns of the black-legged tick, Ixodes scapularis, and Lyme disease in Ontario. Yeah, I'm trying to make a case for a One Health surveillance approach due to the complex nature of the Lyme disease system. So our problem here that we have is that of Lyme disease, which is a vector-borne disease which is rapidly emerging in Canada. The rapid emergence is accelerated by the warming of the global climate and changes in the patterns of land uses. The main vector of the Lyme disease is the black-legged tick, mainly in Ontario and Eastern Ontario. It is the Ixodes scapularis, which is a black-legged tick. In Western Canada, we have Ixodes pacificus. So our focus here will be on Ixodes scapularis. And the range expansion of this tick is evident because since 1991, only one known established population of the tick was found in Long Pine, Ontario. But presently we have numerous endemic regions all over the country, and we have them in Ontario, Quebec, Manitoba, Nova Scotia, and New Brunswick. The range expansion that we are seeing is also coinciding with an increase in the number of cases of Lyme disease in humans and in animals, especially dogs. This risk of Lyme disease has been shown to be presently in all Canadian provinces, but the risk remains highly variable from region to region due to important differences in social and epidemiological contexts, regional environments and landscapes, especially climate variables. So an important question that we have to ask, there are a couple of important questions that we have that needs very specific answers. Importantly, we are asking, how can this rapidly changing environmental risk of Lyme disease and of landscapes be monitored and tracked in ways that the regional differences can be understood? Again, we have ticks in the environment, and these ticks in the environment constitute risk. But how can this environmental risk be linked to human and canine disease incidence in different regions, which are all at different stages of disease emergence? And again, if we have to design risk reduction interventions, how can we do this more effectively in a different context, epidemiologically and environmentally? To answer these questions, specifically, we need to collect data. And to collect the data, we need to use effective surveillance methodology. But even though we are going to use important effective surveillance methodology, other pertinent questions are going to arise, particularly which surveillance method is the most effective or the most ideal? Is it passive surveillance that we will collect ticks from people or from animals and submit them to testing? Is it active surveillance that we are going to collect ticks from the environment? Is it monitoring human cases of Lyme disease, or is it the monitoring of canine cases of Lyme disease? The answer to this is very complex because none of these surveillance approaches can answer all surveillance objectives, and therefore we need more effective methods of surveillance. Unfortunately, the Lyme disease system is quite complex, very, very complex. There are ticks in the environment. These ticks live on hosts. Some of these hosts are reservoirs for pathogens. When ticks bite humans, they cause human disease. When ticks bite animals, they cause animal disease. There's a changing environmental, social, and epidemiological context of Lyme disease. And because we have human, environmental, and animal context, there is the involvement of multiple stakeholders, which bring different perspective to surveillance. They have different practices, they have different training, and all of these make the Lyme disease system more complex, and as well increases the complexity of disease surveillance. So what is the solution to this? The solution to this, we are proposing an adaptive surveillance which can be tailored to the needs of various regions. Regions at different stages of Lyme disease emergence will have different needs of surveillance, and therefore we need an adaptive surveillance methodology which can be tailored to these various needs. The One Health approach, which recognizes the relationship between humans, animal, and environment, is what we propose, which has shown various benefits, especially in generation of early warning signals and in reduction of the cost of surveillance. However, mostly in Canada and in Ontario principally, the feasibility and the utility of this One Health approach has not been documented. And therefore, there's this need for us to document, to show evidence of the utility of the One Health approach. As such, data that we collect from surveillance activities must be used to show that there is Lyme disease risk present in the environment, and this risk is concurrently threatening the human and animal health. This is a premise for our research and the premise for this project. So the project that we are presenting here today has the goal of assessing the temporal trend, that is time trends, and the seasonality of Lyme disease and the blacklegged ticks. We also want to show the spatial distribution of these blacklegged ticks and Lyme disease in Ontario. Specifically, we will monitor the trend, or we will assess the trends using yearly counts of Ixodes scapularis, which are from active and passive surveillance. We will also use counts of human and canine cases of Lyme disease in Ontario. The next objective is to determine the spatial distribution of ticks that are infected with Borrelia burgdorferi, which is the causative agent of Lyme disease. We also will monitor or see the spatial distribution of the rate of submission of ticks from passive surveillance. We also look at the spatial distribution of the human incidence of Lyme disease, as well as canine Lyme disease seroprevalence in Ontario. Lastly, we will determine the seasonality. So once we look at the trend can only tell us if there is an increase or a decrease in these various data sets, but we will not be able to know if within each year we have some cyclical patterns happening. So our last objective is to determine the seasonality, and that is going to enable us to tell if within each year there are certain periods of the year or months that we have more cases or less in Ontario. For us to do this, we sourced data from Public Health Ontario and from IDEX. So there are 4 data sets that we used. The first one is the human Lyme disease case data, which was from 2010 to 2019. It's a huge data set, so it was cleaned, and we included only the probable and confirmed cases of Lyme disease. Cases that were caused by Borrelia burgdorferi were the only ones that were included, and this is because other species of Borrelia are usually travel related. We also included cases only if the public health unit of the individual was identified, because we wanted to show this spatial distribution of those cases. So wherever we didn't find the public health unit, either from the postal code or the forward sortation area, we did not include that case. The next data set was the Ontario passive surveillance data, which was again from 2010 to 2019. We only included the ticks that were Exodus scapularis, and we included a tick if the postal code was used to identify the public health unit from which it came from. The next data set was the active surveillance data from Public Health Ontario. This active surveillance started around, actually we have data from 2014, but 2014 data was excluded because it didn't cover enough public health units. So from 2015 to 2019, we had some very good data that we used. Again, we included only Exodus scapularis, and we used Exodus scapularis from public health units that were identified. The last data set is the animal testing data from IDEXX. IDEXX is a reference lab for testing most of the animal diseases. So from 2007 to 2016, we obtained this data, and we included only the canine serologic testing data. The data set was as well very huge because it contained testing results from all other animals, and so we had to filter it to include only the canine data. And again, public health units that were identified are those that we had data from based on the postal code that was reported. The data analysis that we did were threefold. So first, we did a time series analysis, and then we modeled the time series, and then finally we did the spatial distribution mapping. For time series analysis, they followed first of all an exploratory approach. So I will give you a background of how time series or why time series was done. All data sets, all 4 data sets, were collected sequentially in time from surveillance activities. So from month to month or from year to year. And then these data sets therefore constitute a time series because they have been collected sequentially in time. Why did we do a time series analysis? We have mentioned in the objectives that we wanted to see the trend, we wanted to see cyclical patterns, and only time series analysis will allow us to see these trends and cyclical patterns. So time series analysis will allow us to describe and model this change over time. Because we have 4 different data sets, we wanted to be able to compare how these cyclical patterns or changes have occurred among these 4 data sets. So the time series analysis will also allow us to compare the different phenomena within one subject. In this case, in this project, our subject is the province of Ontario, and the different phenomena that we are trying to monitor are the different surveillance data sets. That is active tick surveillance data, passive tick surveillance data, human case data, and animal case data. So exploratory data analysis was done, time series analysis. Here we wanted to find some general patterns in temporal variation. So just here we did graphical methodology. But then for us to understand how this was done, I'll just give you a background of what data is all about. So the data set that we have will include a signal. Data has a signal plus a component that is noise. So these are just data aberrations. We are focusing on the signal. So the signal will have a trend, a season, and an autocorrelation component. The season here, those are long-term shifts. Like we are saying, we wanted to see trends over time. So those are long-term shifts or changes in the expected value for each data set. And the season is cyclical variations around this trend. So how within each year, are there some variations in this year, and the trend will just give you the long-term shifts. Autocorrelation here will refer to the phenomenon that the observations, that is values of a phenomenon that are collected close to each other will resemble one another than those that are further apart. So with this in mind, we went ahead to do our exploratory time series analysis. Like we said, there is an autocorrelation component in the data that is dependence, either spatial dependence or temporal dependence. These data sets were all checked for this phenomenon of autocorrelation using a test that we call the Dublin-Watson test, a G statistic. So wherever this test was significant, it was significant in all 4 data sets though. Whenever it is significant, it meant that there is autocorrelation or some spatial or temporal dependence in the data. Next, we plotted a time series of each data set. The time series is just a line graph, so it's a linear regression model of the counts of the phenomena against time. So in this case, it was count of exodus scapularis that was collected from active surveillance against time, counts of exodus scapularis from passive surveillance against time, counts of human cases of Lyme disease against time, and the counts of canine cases of Lyme disease against time. The time series plots will only give you a graph, but we wanted to see if there's some cyclical variation or there is some trend. And to do that, the time series graph will not generally give you that. So we had to decompose the time series using the STL methodology. Next, we did regression modeling of the time series. Like we mentioned, the time series plot will only give you a trend, even if you decompose. So at best, with the time series plot, you could only describe if there was an increasing trend or there was a decreasing trend or no trend at all, and if there is some seasonal effect. But we wanted to quantify by how much there has been a trend, by how much there has been an increase, and actually see a plot of the seasonal effect. So we did a model of the data of the time series data. So this model was the generalized linear autoregressive moving average model. The model allows us to model the trend and the seasonal effects together. Other models such as the ARIMA or SARIMA models will integrate the trend and the season. And when they do that, they will filter out the trend and season. And at best, you can only describe them. You cannot quantify. So the GLAMA models here allow us to actually allow the trend and the season in the model. And so we'll be able to describe and explain and quantify them together. The trend here was modeled as a 1-unit increase over the entire study period. And we centered this trend around the mid-study period. And for the season, normally we can use the months of the year as season, but then we have 12 different months. And if we put all of those in the model will become too cumbersome to interpret. One of the months will serve as a referent, but there are going to be 11 other months that we will need to explain. So in modeling, the best approach is usually to have a model which is very parsimonious. That is very simple with the least number of variables in it. Therefore, the season year was modeled as an annual and semi-annual sine and cosine harmonic functions, which allow us to have just about 4 or 2 variables which we can easily explain. For the models, we did the Poisson and negative binomial models. Yeah, here we have count data, and because we are having some kind of serial dependence in the data as shown by the Dublin-Watson test, we decided to use the Poisson and negative binomial models. So for each data set, we have 4 data sets. For each of them, we built 18 different models. And these models were compared using the archaic information criteria, AIC. The smallest AIC, the model with the smallest AIC was selected for further investigation. In the case where we had a positive or a significant negative binomial dispersion parameter, we selected that model over a Poisson model because that meant that there was over dispersion in the data set, and therefore a Poisson model will not be a best fit. We applied the model diagnostics to selected models, and those enabled us to see if the model fitted the data or it did not fit the data. Now, there was one other thing that we did. The GLAMA models they utilize an AMA structure. And these are also linear models, but those linear models will allow us to model the trend and the season component in the data. The general linear models will just serve as a linear regression model. So in order to check if the GLAMA model was superior over the GLM, we did a likelihood ratio test. And the null hypothesis for the likelihood ratio test here is that the model is a GLM. Wherever we had a p-value which was less than 0.05, it meant that we had enough evidence to reject the null hypothesis that the model was a GLM, and so we accepted the GLAMA model. Finally, we mapped the data using the Public Health Ontario Easy Maps tool. This can be obtained from this link, and this is online, is available, and is very easy to use. We just uploaded a data set that we have based on various IDs from health units, and we obtained the various maps that we wanted. So after our analysis, we had from 2010 to 2019, there were 29,291 Exodus capillary sticks submitted from passive surveillance. That gave us a submission rate of about 214 per 100,000 human population in Ontario. Out of those sticks, 25,819 were tested for Borrelia, and 4,625 were positive for Borrelia burgdorferi. Infection prevalence was 18%, and the mean monthly submission rate was 244 ticks per month. So, as indicated, the first thing we did was a time series plot. Here again, you can see that this is a time series plot for the passive surveillance data for ticks, but you can't clearly see if there's a trend or there's a seasonal variation. So we had to decompose the data, the time series. The decomposition here shows us that there was a slight increase from 2010 in the number of ticks submitted from passive surveillance, but that was a dip in 2015. Yeah, at this point, some public health units in Ontario had stopped the collection of ticks from people, from the public, and we think that that is why there was a dip in the number of submissions. But after 2016 until present, we are seeing that there is an increase, a constant increase in the numbers of ticks that are being submitted. A GLAMA model that we did allowed us to show that there is a positive correlation between the number of ticks and in time. That model, I didn't show yet the results of that model, but wherever we have the coefficients of a linear regression model being positive, that shows that there's a positive correlation. And in all the 4 data sets, the GLAMA models, the coefficients of the trend were positive, and therefore that showed us that in all of the 4 data sets, there has been an increase in the submission rates of ticks, as well as the cases of human and canine Lyme disease. So from the GLAMA model, we did plot the seasonal effects. And clearly here we can see that for the passive surveillance data for ticks, there is a peak, there's a bimodal distribution with a peak first in May and again around October. This is shown or has been shown in a lot of studies, and again in Ontario we are seeing that it's following that same pattern. This has implications that we are going to discuss later on. From mapping, I'll draw your attention here to the fact that the phenomenon for passive tick surveillance, the rate of passive tick surveillance in Ontario, much of that is happening around the eastern parts of Ontario. You're going to see that in all of the 4 data sets, the eastern part of Ontario is being highlighted, and we are going to have discussions towards that. For active tick surveillance, that is from tick dragging, environmental sampling, there was a total of 3,067 ticks submitted between 2015 and 2019. They tested 3,065 and 890 were positive for Borrelia burgdorferi. Infection prevalence here is 29%, which was or is somewhat the same or similar to various analyses that we are doing right now. from active tick surveillance and from passive tick surveillance in dogs. I just finished one of the studies that I did. And the surveillance, the positivity rate is as well around between 23 to 29%. So the time series plot, here again, we cannot see any pattern, but if we decompose the time series of active tick surveillance, it shows us here that there is a constant or there's an increase in the numbers of ticks that are being found in the environment from active tick surveillance, which mirrors the same phenomenon that we are seeing from passive tick surveillance. And again, the seasonality is showing us the same pattern that we had for passive tick surveillance. There are more ticks being found in the environment. The peak is in May, and again, it is in October. So here we have passive tick surveillance ticks from people and active tick surveillance ticks from the environment having the same seasonal patterns. And again, like I said before, there's a lot more of it happening, more of the broiler bug diphtheria prevalence, infection prevalence in those ticks from active surveillance is happening in the eastern parts of Ontario. I want you to note here what is happening in the northwest of Ontario. This has implications for One Health and the generation of early warning signals, as I'm going to discuss later on. So in eastern Ontario, we are seeing a signal here for the infection prevalence of broiler bug diphtheria in Ixodes scapularis ticks. Human Lyme disease case data. From 2010 to 2019, there were 4,603 cases of Lyme disease reported. So this would be probable and confirmed cases based on case definition. The incidence rate was 33.5 per 100,000 population. We have 3,921 confirmed cases as against 682 probable cases of Lyme disease. The mean monthly case number was 38. So we had 38 cases per month over the study period. Again, here we have the time series plot. We cannot find any patterns, but if we decompose the time series, then we have a trend. There is an increasing trend that we have seen for the past two data sets. The number of cases of Lyme disease in humans is also increasing over the years. The cyclical pattern, however, is a little bit different. We see that there are more human disease cases occurring. So the peak year is in July. This is because the average, somewhat average incubation period of Lyme disease is around 21 days to 2 months, 3 months. So there is going to be a lag. Once in the other two cases, we had peaks occurring in May, and now we are seeing the incubation period of Lyme disease is around 21 days to 2 months. So you're going to have an increase in the number of cases after like 2 months. And that is why we are having more of the human cases occurring in July. Again, if we plot this to show the distribution of human incidence of Lyme disease per 100,000 population, we are seeing again the signal that is occurring in the eastern parts of Ontario, just like we had in the passive surveillance data and active surveillance data. The canine Lyme disease serology data from 2007 to 2016, we had 719 dogs, 719,026 dogs tested. Out of those, 12,677 dogs were positive for Lyme disease. And then we have a seroprevalence of 1.7%. This is very similar to a lot of work that has been done around Ontario, usually ranging from 1.2 to around 2% seropositivity or seroprevalence rate. So we think that this is something that is plausible. The mean monthly count of seropositive dogs, so there were 106 dogs per month positive for Lyme disease during the study period. The time series plot, again, here we cannot find any kind of pattern, but if we decompose this graph using the STL methodology, we see that, again, here, this is a fourth data set from canine cases of Lyme disease. There is an increase in the trend. So we have more positive dogs over time for Lyme disease. And the seasonality, we are seeing here that there is a peak as well in May, which corresponds to the peak of tick submissions from passive tick surveillance and from active tick surveillance. However, there's a caveat that I want you to note here is that in the spring of each year, dogs, veterinary clinics receive more dogs and they test more dogs because dogs go for screening for heartworm disease. Heartworm disease is also a tick-borne disease, and usually the test that is done for those dogs is a 4DX test that is from IDEX that I know of, that the data that we're using here. It's a 4DX test, and that 4DX test tests for 4 pathogens. One is the heartworm, then anaplasma, and then Borrelia burgdorferi and Escherichia canis. So in that case, we are seeing that most likely the pattern we are seeing here could be because more ticks are being tested more dogs are being tested in the spring, but as well, we can confirm here that the peak of Lyme disease is the peak of ticks in the environment and from passive tick surveillance is occurring around this time. And so we need to note what is happening there. If we map seroprevalence of Borrelia burgdorferi in dogs, again, we are seeing these signals in the eastern parts of Ontario, and we can see what I've said about active tick surveillance here in the northwest region of Ontario. I'm going to explain or talk about that in more detail. So we have shown from all the STL decompositions that there's an increase. I just wanted to bring these graphs on here so that you can appreciate exactly what we have been showing all along. So in all 4 cases, for the human cases, the seropositivity in dogs, the counts of active tick surveillance, and the counts from passive surveillance, we are seeing that all 4 data sets are showing an increase over time for the various study periods. Again here, for the seasonality, we see passive tick surveillance and active tick surveillance. The counts of the ticks from those are picking up in the same months. And we can as well see that the cases of disease in humans and animals are almost picking up in the same months. So again, I wanted to show here the different maps for active, passive, human cases and canine cases of Lyme disease. We can see here, the first uppermost part is the human Lyme disease cases, which is corresponding to what we have here in the rate of passive tick submissions from humans. The rate of passive tick submissions from humans in other studies have also been shown to correspond to the number of Lyme disease cases that are occurring in those regions. However, what I want to draw your attention to is the fact that we have here, this is the map for the active tick infection prevalence. As here is the map for, on the upper right hand is the map for seroprevalence of Borrelia burgdorferi. It shows the same pattern. And again, we are seeing a signal that is generated in the northwest region of Ontario for both the active tick surveillance and the canine seroprevalence. This signal is not shown or is not being generated in case of the human case data, as well as from passive tick surveillance. Therefore, it is suggesting that dogs are picking up signals earlier than what humans are, what we are seeing in humans. And to confirm this, we can clearly see that the data set for the seroprevalence data is from 2017 to 2016. And the data set for human and passive tick surveillance is up to 2019, which means that earlier on, since 2016, we are already seeing some phenomenon happening in dogs in the northwestern region of Ontario. And that is also shown for the active tick surveillance, so environmental surveillance happening still in that region. This tells us that probably dogs are picking up signals earlier than humans, and so dogs can serve as effective sentinels to provide us with early warning signals of tick invasion in an environment, as well as the cases of disease. Now, in the One Health system, if we did not bring all these 4 data sets from the environment, from humans, and from dogs, we would not be able to make this conclusion. So we can clearly see here that there is a concurrently increasing trend in the monthly counts of human cases, tick submissions, and canine seropositivity, as has been shown in our various graphs. We can see that it's an increasing trend. This trend is concurrent for all of the 4 data sets. The seasonality as well is somewhat concurrent. As well, we are seeing a concurrent distribution, spatial distribution in this case. We also have seen that there is marked seasonality, which was observed in the cases of tick counts from both the passive and active tick surveillance. Both peaks are in May and in October. So that tells us that once ticks are found in the environment, they are likely to start biting humans, and this is occurring during the same periods of the year. And these are periods where you have more of the ticks happening present in the environment. Data has shown that if these ticks, if we checked at the stages, we would have more of the names happening here in the earlier months of the year, and the adults. Part of my research has shown that most of the adult ticks are present or are occurring in October and November. The next conclusion is that most of the human cases were reported between May and August, and there is a peak in around July. So we have here human cases. Most were reported here between the May and the August period of October period, and then there is a peak here in May. This peak coincides as well with the peak in seasonality of seropositive dogs. Next conclusion is that the incidence of human cases of Lyme disease and all the rates of submissions and the ticks were all present more in Eastern Ontario. So the Eastern Ontario part is where we are having more of these signals happening, and other risk maps that have been generated over the time have shown this same pattern. We also conclude here that broiler bug-the-fly infection prevalence in ticks from active surveillance, canine prevalence were all highest in Eastern Ontario and in the northwestern region of Ontario. The phenomenon that I wanted to show you again is, just like I've explained, that there is some signal that is being picked by dogs earlier happening in northwestern Ontario and active surveillance. So active surveillance is giving us ticks present in the environment, and dogs are already picking these signals earlier before disease happens in humans and before ticks start being submitted from humans. Now, what is the next question for us? The next question for us is to know by how many years or how long does it take for ticks to arrive in a region, and when ticks are going to be picked by humans and then human disease. My next projects are going to be showing this pattern when we are going to do modeling to show that there is a period of time from the time when ticks arrive to the time when we start seeing human disease. There's a next project as well, which is almost concluding, that will show us that dogs are picking up signals earlier. The work that we have done here now is from historical data. And so we are now actively collecting data to prove that this is what is happening in Ontario. So in conclusion, our findings have confirmed that there is the concurrent temporal and spatial aggregation patterns of Ixodes scapularis in the environment. That is from both active and passive surveillance. There is this concurrent nature of human Lyme disease incidents and canine Lyme disease in Ontario. Therefore, we have brought up these findings based on research that has been done or from analysis that have been done using environmental data, that is from active surveillance, human case data, passive tick surveillance data, and canine Lyme disease data. So we have both human, environmental, and animal context or components of the data sets that have been combined to give us this. However, if we check, we would see that the data sets have been collected over different time periods, and that's a challenge for data integration. However, even though these data sets have been collected for different periods of time, integrating them have given us some very important signals, which means that the One Health approach, which we are advocating, to collect data from environments, humans and animals is going to be the best method, because information from one system or from one component is going to help us to determine if there is a risk for the next component. So for example, as we mentioned before, dogs are picking up signals earlier than humans. Can we utilize dogs as important sentinels to inform that there's going to be human cases of Lyme disease in an environment? If we have ticks in the environment, what is the probability that over a certain period of time, we are going to start having human cases of Lyme disease as well as tick bites in humans? So this One Health approach is what we are advocating for, and we think that the One Health approach is going to help us or enhance our ability to properly or effectively monitor and prevent and treat Lyme disease and other tick-borne diseases in humans and animals in Canada. However, we think that the stakeholders that are involved in these systems need to come together. It is a very big challenge for collaborations to occur. And so one of the projects that we have done is to verify if stakeholders are, first of all, willing to participate in this One Health approach. We have seen that from that research that stakeholders are willing to participate in the One Health approach. Now we have asked what are the needs and challenges that they may face. We now know all of this information, and the way forward is for us now to design. That is my next and final project, to design a framework of how the One Health surveillance system is going to be done and how it's going to be established and how we can evaluate it to show that it is effective and how well it can be adapted to different environmental and epidemiological contexts. With that, I thank you all for listening, and I'm open to questions.
Thank you so much, Cyril, for your informative presentation. We are going to move now to the Q&A segment. I'm just looking at the chat box right now. I don't see any questions yet, but if anybody from the audience would like to ask Cyril a question, please raise your hand icon or unmute and ask your question. Just looking and seeing, and forgive me if you already mentioned it, Cyril, but how many regions as part of your canine surveillance program that you piloted last year did you roll out?
The canine surveillance, we have done that for 4 regions already. Those 4 regions are Hamilton, Guelph, Muskoka Lakes, Gatineau, that is Ottawa, Gatineau, and Kingston area. So those, we have the data coming in right now. We have done the first phase and the second phase is now going on.
And are you expanding the number of regions that or are you keeping it at the same 4 regions?
We are keeping that at the same 4 regions. However, active tick surveillance is going to happen in more regions. We want to pilot this first phase for 4 regions. We will analyze to see what signals we pick up from this phase before we can make recommendations for scaling.
Perfect. Thank you so much. We still have time for questions from the audience. Don't be shy. Please raise your hand icon up. Here we go. Dr. Kolati, please go ahead with your question.
Thanks for that presentation, Cyril. That was really interesting. I was really struck with your point about finding Borrelia in the northwestern part of Ontario. And it made me think about other tick-borne diseases or other pathogens carried and how dogs might be a sentinel for those. Do you know what else Public Health Ontario is testing for in these ticks for the data you were using?
We are testing as well for anaplasma. I think Public Health Ontario is not testing for a lot more pathogens except the National Microbiology Lab. But I know we are testing for anaplasma, we are testing for Borrelia miyamotoi and other Powassan virus and Babesia. I'm not very sure if we have those results, but I think from the data set, we have results sparingly for most of these. I know for anaplasma, I know for Borrelia miyamotoi, I know for Powassan virus, Babesia micro-T, but those results are not very, very consistent. We are not having the whole data set for those. And actually, I have some of the results for these pathogens, but we have not done any like analysis to show that there is some any kind of relationship. The best we have done is for Borrelia burgdorferi, which is what our focus is on.
Right. I guess those are pretty rare still for the
Right.
most part. Yeah. Thanks.
Thank you, Dr. Kolati, for your question. We still have time. Does anybody else from the audience have a question for Cyril? I'm just scrolling up and down. I don't see any additional questions. We have a quiet audience today for you. I'm just doing one last check. If anybody has a question, please raise your hand or unmute and ask Cyril. This is your opportunity. He's going to be very busy soon. Oh, go ahead, Dr. Morshed.
Yeah, it's kind of giving some tough time to Cyril. So I think it was an excellent presentation, but you know that we have many diseases and many of them are high profile. And Lyme disease, so far my understanding, is not that high up on the agenda, although many people are suffering significantly. With the limited number of cases, the proposal that you gave, all sorts of things to have a One Health approach like active surveillance, passive surveillance, clinical diagnosis, this and that, you know, surveillance costs money. And we are doing it. It was very tough to get okay from the admin folks to go ahead, but still we are managing. But I think that we are the only province is doing passive surveillance on a continuum basis. But from Whitehorse to Newfoundland, Labrador, very few areas they are doing that. And then mostly my gut feeling is the money is the number one stumbling block. So what would be your thought process that we can do something magic, then we can do the passive surveillance on a continuum basis and do some sorts of disease identification like a panel or something, so that we have a kind of idea that when the things is going up or down or something. So what is your thoughts on that?
Thank you, Dr. Marshall, for this. That's a tough question, and usually that is where the One Health buy-in comes in. Like, what my thought here is that the One Health approach is not only going to be applied to vector-borne diseases. There are a whole lot of other health problems that can be solved using a One Health approach: antimicrobial resistance, food safety, and stuff like that. So the framework that we want to build, or that I'm going to build, that is going to make recommendations, not just for Lyme disease, like you said, is low profile. COVID-19 is there right now, and it follows the same kind of pattern with Lyme disease, involving animals, environment, and humans. So most of what I'm going to do is to show that this approach can be utilized in every context and not just for Lyme disease. And should we apply this or accept the recommendations, it is going to help us to monitor other diseases and not just Lyme disease. The next thing is that this is the most difficult part of Lyme disease of One Health. It is to show that added value that you can obtain using the One Health approach. So collecting data from passive surveillance and combining it with the rest of the data, it becomes very difficult. But then if we can begin to show that we are picking up signals in animals earlier than they are occurring in humans, or we are picking up signals from the environment, then we can very clearly demonstrate that we are capable of using a One Health approach to pick signals earlier before disease start occurring in humans. And in preventive medicine, we want to be sure, or want to make sure that we are picking up these signals, and so we can put in measures that could serve as barriers to human infection. This is a two-way process. one of the issues with One Health is that it is using the anthropocentric. Most of the projects have gone anthropocentric approach, but then there is risk to animals as well. So we want to be able to show that we can utilize animals in their own good and not just for informing human health. There's one of the projects that I am doing, which is going to use human data as a means to predict animal data. So by combining most of these data sets and showing these two-way processes, it increases our chances of having that buy-in from governments and public health departments.
Okay, thank you. And also, I have one appeal to the patient group. I think that some of them are attending, and thank you for attending to this talk. I'll just appeal one thing, that in my long career that I have seen many organizations such as Lyme disease organizations in different parts of the country. So we have, as for example, TB society, we have cancer society, we have HIV society, we have hepatitis society. And what is happening, those societies is Cancer Foundation, as for example, they are collecting or they are campaigning for millions and millions of dollars and giving to the top-notch researcher to do more work on this. So my appeal would be that somehow that scientist, I'm sorry to say, and I can say it now because I'm old, so nobody will give me trouble. So somehow that Lyme disease patients group and scientists or public health scientists, particularly like me, that there are some disconnect. So my appeal that this patient group should be more kind of positive towards the scientist and work together, collect some funding and giving to the young researchers, not old people like me, like young researchers, so they can do a top-notch work to break the barrier that why we cannot diagnose, why we cannot give the right treatment, why we cannot do this. So I think we need to be much closer than where we are right now. So I think that somehow somebody should take the leadership from the patients group and also from the scientist group. So that will be my appeal for today.
Thank you very much, Dr. Maset.
Thank you, Dr. Borshad, for your thoughts. Thank you, Cyril, for addressing the questions. I'm looking here. I don't see any questions. One last call out. If anybody has a question you would like to ask Cyril before we close up today's discussion. Here we do. We have a question. Let me just click onto it. Are there some canine species that are more resistant to Lyme infections? And how does this affect prevalence?
So our work has not explored canine species and how they relate to Lyme disease. I think that one of my colleagues in the lab, what she is doing is looking at that aspect of it, but she's looking at instead the strains of the bacterium causing Lyme disease against clinical infection. However, we are not very sure if the various canine species will be affected differently by Lyme disease. We are not very sure. That I have not seen any research. What has been shown is that various species have different acquisition rates of the ticks. So, for example, the fall length of the ticks of the dogs, or for example, the rate at which this dog is active will determine how much or how many ticks this dog can acquire from the environment. Clinically, we are not sure, or I'm not sure of what happens when the tick has been acquired and the dog has Lyme disease, and how that spans out in terms of disease. I'm not very sure of that.
Thank you for your question, for addressing it. There was a bit of an echo there. Thank you, Cyril, again for presenting today. I'm just going to stop sharing your screen and share my screen to just remind everybody of what's happening tomorrow. Again, Thank you so much, Cyril, for your presentation and spending the
welcome.
time today to answer the questions from the audience. We're back tomorrow for one last presentation this week. At 12 o'clock noon Eastern Standard Time, we have Dr. Seema Afsharnazad, who will be presenting on handheld microbiome sequencing to detect, identify, and monitor tick-borne pathogens. We hope you join us tomorrow for her presentation at noon. Then we will take a break and we're back next week on Monday and Tuesday for our last set of presentations for Lyme disease awareness event. And just a reminder, it's not too late. Please take part in our challenge, wear green or wear a green face mask, take a photo and share it with us. If you don't like your photos taken, feel free to send a picture of any art or creative work you've done that expresses Lyme disease awareness event. All entries received will be automatically entered into a draw for one of 4 $25 Starbucks e-gift cards. And our draw will occur on our last day of our presentations on May 31st. And you can send your entries to clydern@gmail.com. Again, thank you everybody for attending. Thank you for our audience for your questions. And again, thank you, Cyril, for your presentations. Take care, everybody. Have a great day and a great rest of your week.
You do take care. Bye, everyone.
Bye.